Thanks everyone for coming and attending this week's M2D2. This week we have Michelle Lee. So Michelle just defended her PhD in biomedical informatics at Harvard University. In her research, she innovates contextual learning algorithms on graphs to enable precision medicine. Her work aims to tackle challenges of diagnosing patients with rare or even novel diseases and facilitating drug discovery in a cell type specific manner. And today we will learn about contextual AI models for single cell protein biology. Thank you, Michelle.
Thank you so much for the invitation. And yes, I recently defended yesterday. So this is my first talk as a PhD, which is kind of crazy. But thank you again just for being here to hear a little bit about some of the work that I've done during my PhD. And specifically, I'll just share one aspect, contextual AI models of single cell protein biology. So first of all, what is context and why do we need it? So let's take apple, for example. If we were to use it in two different sentences, grow an apple, buy an apple, would we get the same autocomplete recommendations from Google or ChatGPT? No, because apple means different things depending on its sentence. Now, the same is true in biology. So H2AFX is a gene that has many functions depending on its cellular context. Now, this is known that gene expression and protein function depend on cellular and tissue contexts, and biological functions can vary considerably across healthy and disease states. So models that aim to generate predictions on proteins should consider the different types of contexts that they may be operating under. But current protein representation learning approaches, including those that are structure and or sequence-based, are context-free, as in each protein only has one representation learned from a single context or an integrated view across many contexts. Meaning these context-free models are unable to adjust their outputs based on the context in which they operate. And so the question that we're interested in is, how do we develop models that can adjust their outputs to the biological context in which they operate? And so we introduced PINNACLE, a contextual AI model that can enable precise protein representation learning and cell type or cell state specific prediction. I'll first discuss the data, and then I will discuss the model in detail. So first, about the data. To be able to model proteins across cell type contexts, we need cell type specific protein interaction networks, which are these networks that are in the peripheral. To construct these cell type specific protein interaction networks, we start from a multi-organ single cell transcriptomic atlas of humans. In this case, we use Tabula sapiens, which is a large-scale single-cell atlas that contains samples from 15 donors, resulting in about 500 cells from after quality control processing. For each of these cell types, which are classified by this atlas, I get a list of genes that are activated in the cell type. In short, activated means that these genes have higher average expression in the cells of interest, aka the cells that are classified as a given cell type compared to the rest of the cells. And then from these lists of genes, I take the global reference PPI network, extract the proteins corresponding to the activated genes, and then take the largest connected component. As a result, each cell type network has around 2,500 proteins, and there are 156 of these cell type specific PPI networks that span 62 tissues of varying hierarchical scales. Now, to incorporate insights about cell type communication and tissue hierarchy, we need a metagraph that captures exactly these relationships. So we have cell type nodes with these diagonal lines here. The colors represent cell type and identity, and these cell type nodes are given by Tabula sapiens. We also have tissue nodes in gray with these dark blue outlines. The color, the size of these cell type tissue nodes indicate the level in the hierarchy. So smaller means closer to the root node of the ontology, meaning they're more broad. And cell types come from a specific tissue according to Tabula sapiens metadata. So those tissue nodes are connected to the 156 cell type nodes. Cell type to cell type edges means that those cell types interact. And tissue to tissue relationships are indicating parent-child tissue relationships in the tissue hierarchy. Okay, so now that we have a better understanding of our data, let's talk about PINNACLE. So first, let me give you an intuition of the model. So given our cell type specific PPI networks and the metagraph, PINNACLE embeds the proteins, cell types, and tissue nodes in a single unified embedding space, such that the proteins from the same cell type network are attracted to each other if those proteins share an edge, as in they're closer together in the embedding space. If they don't share an edge, then their embeddings are repelled from each other, as in they are farther away in the embedding space. Protein embeddings from the same cell type network are attracted to the corresponding cell type embedding and repelled from other cell type embeddings. And cell type and tissue embeddings are embedded nearby if they share an edge in the metagraph, otherwise they're repelled. The goal here is to learn an embedding space that is organized according to the behavior of proteins at each cell type based on network topology and interactions between cell types and the hierarchical organization of tissues. So let's go a little bit deeper into how this is mathematically formalized. So there are three key components of PINNACLE. There's the protein level attention mechanisms with cell type specificity, metagraph attention mechanism on cellular interactions and the tissue hierarchy, and an attention bridge between the protein and cell type embeddings. So first up is the protein level attention with cell type specificity. So the superscript PP here means that we're dealing with protein-protein edges. So for each node, we aggregate the neighboring nodes embeddings H with an alpha attention mechanism alpha and a protein-protein edge type specific transformation matrix W to project the features of the proteins in the cell type specific PPI network. And there's one W matrix for each cell type specific PPI network. This similar message passing is used in the cell type and tissue nodes in a metagraph as well, but separated by edge type. So there's cell type to cell type to tissue, tissue to tissue, tissue to cell type. And then we aggregate these different views of a node's neighborhood by learning an attention over the edge types. Now, finally, you may have noticed that there are no edges between the proteins and the cell types. So to allow messages to be propagated between messages and proteins and edge types, we have an attention bridge here called gamma. And this means that to learn a cell type embedding, we take an average attention weighted average over the proteins that are activated in the cell type. And the same gamma is used to regularize the cell type specific protein embeddings. As for our objective function, we have three terms. We have loss at the protein level, cell type level, and tissue level. So at the protein level, we perform link prediction on the PPI edges. We also predict which cell type the protein is activated in. At the cell type and tissue levels, we perform link prediction only, but considering separately the different edge types. For link prediction, we use binary cross entropy, pretty standard. For cell type classification, we use center loss. As a result, we have a single unified embedding space where protein representations are organized according to cell type context and tissue organization. So in these UMAP plots here, you'll notice that the proteins from the same cell type context are enriched in a particular embedding region. Separately from this UMAP, we directly use an approach called spatial analysis of functional enrichment on PINNACLE's embeddings to show that indeed these spatial regions in PINNACLE's embedding space are significantly enriched for protein representations originating from the same cell type context. So altogether, PINNACLE provides contextualized protein, cell type, and tissue representations that we can now use to finally generate contextualized predictions with cell type resolution. So given our contextualized protein representations, we can now fine-tune them for any downstream task of interest. In this case, we're interested in predicting whether a given protein is a strong candidate for a therapeutic area of interest. This is actually a binary classification task. So given a protein, is it a strong candidate? 1, yes, 0 means no. But because we have unique protein representations per cell type context, we can additionally gain insights about which cell types are highly predictive to examine therapeutic targets in the area that we're interested in. So how do we define protein targets? First, we have our universe of druggable proteins and our protein-protein interaction networks. These are proteins that are nominal targets of existing drugs. A subset of those proteins are proteins that have been studied in our therapeutic area. A subset of those have been investigated in clinical trials. And a subset of those have been investigated in clinical trials that have at least completed phase 2 of clinical trials. And this becomes our positive chain examples, as in they have a label of 1. And this subset of druggable proteins that have not been studied in our therapeutic area of interest becomes our negative chain examples. So the 2 therapeutic areas that we choose to study are rheumatoid arthritis and inflammatory bowel disease. Since there are subcategories of RA and IBD, we broadly define RA and IBD by including these diseases here. We curate clinical trials for these diseases and subset to those that have at least completed phase 2, and these become our set of positive training examples. When we fine-tune Pinnacle to generate contextualized representations, we find something incredibly exciting here. So on the x-axis is our performance metric, and on the y-axis are the different models that we evaluate. So the first row here is Pinnacle. Each dot represents a cell type context. So you'll notice that there is a spectrum in the performance of these cell type contexts. This suggests that there are some cell type contexts that are better at predicting therapeutic targets for RA and IBD than others, which is consistent with what we would expect. Now, these next 3 rows are baseline models, all of which are context-free. From these models, we only see 1 data point shown by the square because these approaches can't adjust their outputs depending on the context. So you'll notice that for RA and IBD, there are many cell type contexts that outperform these baseline models. This means that Pinnacle's representations predict drug targets for RA and IBD significantly better in these cell type contexts than the context-free methods. So let's take a look at what these cell types are and why there are some cell types that do not perform better than the baseline models. So here's a zoomed in version of the performance for RA, except now the performance is on the y-axis and the cell types are on the x-axis. So the dotted lines here represent the baseline model's performance. Again, each dot here represents the performance of proteins from a single cell type, and we've validated with an RA clinician, Catherine Liao from Brigham and Women's Hospital, that the top ranking cell types are highly relevant to RA and align with the literature. And the low ranking cell types are not so relevant for to RA. We can also look into individual proteins and examine the most and least predictive cell types to better understand their mechanism of action. Yes, I see a question.
Yes, I was just going to ask, what's the baseline here?
Yes, so the baselines here, I'll just go back to this previous slide. So they are bionic. So I have 3 baselines here. So it's bionic, GAT, and random walk. So bionic is a set of graph convolutional layers that integrate the cell type specific protein interaction networks to generate a single representation for each protein. Graph attention neural network, or GAT, is a learns on the global reference PPI network to generate a single representation per protein. And then the random walk is a diffusion-based approach that operates on the global reference PPI network as well. Great question, thank you. And so this brownish dotted line here is bionic. This gray here is GAT, and then I don't include the random walk here because it was essentially 0. So yeah, awesome. And when we look at IBD, we see similarly exciting results. And we've worked with Dr. Ashwin from Mass General Hospital to verify our findings here. And again, we can look at these individual protein level predictions across these different cell type contexts to better understand their mechanism of action. Another aspect that's really important for therapeutics is predicting protein-protein interactions. So for example, in immuno-oncology, interactions like PD-1 and PD-L1 and B7-1 and CTLA-4 are key players in cancer immunotherapies. But our 3D molecular models of these proteins are not necessarily context-specific. So we were curious whether injecting context using our contextualized protein representations would help us better differentiate between binding and non-binding proteins. So what we did here was we took our protein representations generated from 3D protein structure and concatenated with either a context-free embedding or a context-aware embedding. So the context-aware embeddings are Pinnacle's protein representations, and the context-free embeddings basically is an average of Pinnacle's context-aware embedding. So we're essentially averaging out the context. And we calculate their binding scores, which are cosine similarities between a pair of proteins. So on the left here are results from context-free representations, and on the right here are contextualized representations. The black and dark purple dots represent the average score of the binding proteins, and the light gray and light purple dots represent the average score of the non-binding proteins. The important comparison here is the gap between the dark and light colored dots. So with the context-free representations, the gap is 0.006 and 0.003. With the context-aware embeddings, the gap increases to 0.011 and 0.015. And yes, the gap here is quite small, or the differences are quite small, but we do find that they are statistically significant. So to summarize, Pinnacle's contextualized representations are contextualized based on cell type context by integrating multiple context-specific data and learning context-specific importance. It also transfers knowledge across cell type-specific networks, as well as from proteins to cell types, tissues, and so on. And altogether, this allows Pinnacle to perform diverse tasks across cell type contexts. Now, since we've shared Pinnacle with the community, there's been a lot of excitement surrounding the utility of contextual learning. Of course, we're naturally interested in scaling up Pinnacle to a much larger cell type single cell atlas to enable even more context-specific tasks. We've also started a collaboration with ASAP, Aligning Science Across Parkinson's, which is a global initiative to study Parkinson's disease to prioritize cell types based on PD relevance. And we're really eager to work with the wet lab scientists to experimentally validate Pinnacle's hypotheses about which cell types to examine certain PD genes in. We're also following up on our exciting results produced by Pinnacle for RA to improve model accuracy and to enable the model now to perform patient-level predictions. So I'll tell you a little bit more about RA Pinnacle here. So as you may recall, Pinnacle is trained on healthy individuals' single-cell data. So we're working on improving performance on predicting RA targets by retraining Pinnacle on this synovial single-cell atlas from patients with RA. Now, because this atlas also has matched patient single-cell data and clinical data like their RA stage, their treatment history, and treatment response, we can also extend now Pinnacle to generate patient-level predictions. And this is really key because before, Tabula sapiens didn't have some of the key cell types that are known to be, that have been established as being important for studying RA. Of course, we had all of the immune cell types that are involved with RA, but not the synovial cells, which are enriched here in this cell type atlas. So we're particularly excited about being able to now actually evaluate on synovial cells to try to find the most relevant cell types to now do perturbation experiments, to correlate with the clinical data that we have on patients, etc. There's a lot of potential now to incorporate context to generate contextualized predictions for precision medicine. For example, I'm really interested in, given a patient in scenarios where we have a patient who has a loss of function in a particular mutation, and with an understanding of that particular gene cell type specific activity, and our contextual AI model like PINNACLE or a variant of PINNACLE, we can make recommendations like consider compounds whose mechanism of action upregulates this particular gene in this cell type. Or say, given a perturbagen of interest that targets a particular gene, and our understanding of cell type specific knowledge and our contextual AI model, we can predict which cell types we may observe off-target effects in and why. And I think these are really exciting times for single-cell biology and AI. And while I agree with this article's position that we aren't quite at a point where single-cell analysis can be integrated into routine clinical practice, I do think that PINNACLE models can bring us one step closer. That is, if we define precision medicine as being able to accurately match an individual patient to the most beneficial therapy based on the patient's genomic and molecular profiles, contextual AI models like the follow-up variants of PINNACLE have the potential to enable exactly that, adjusting predictions given the patient's context. I even believe that contextual AI broadly can realize the promise of precision medicine. Another aspect of my PhD research that I am not spending the time to talk about today is innovating contextual AI models for rare disease diagnosis. I've developed this model called Shepherd that can perform multifaceted rare disease diagnosis by modeling patients as subgraphs in a rare disease knowledge graph to learn patient-specific context. And so Shepherd can be used at many aspects of the diagnosis pipeline. So after the clinical workup, after genomic analysis, and after case review. With just a list of the patient's phenotypes or symptoms and our rare disease knowledge graph, we can perform causal gene discovery, identify patients like me with the same causal gene or disease, and provide interpretable characterization of novel disease presentations. And with that, I'd just like to acknowledge the lab and our funding sources and here are some resources if you're curious to learn more, and I'm very happy to take questions now.
Great for the talk. Yeah, if there are any questions, feel free to mute, type in the chat. Maybe to get us started, I'm curious if, you're talking about scaling earlier and getting more data. Do you think those models can somehow be used to guide us in terms of where do we need more data, what type of data, and all that?
Yes, that's an excellent question. So this is perhaps an outdated slide by now because the data set that we're scaling to is much larger. But we are really interested in now trying to model more cell types and more tissue so that we could, first of all, better understand the kinds of diseases that we could study now at this stage, given the data that we have, what kinds of hypotheses that we can generate, and even what data do we still need, as you're insinuating. And so I absolutely think that we could get to a point where we can have these conversations with experimental scientists and clinicians and try to really understand where the gap is in their workflow and try to help provide those hypotheses. And if we're not able to generate sufficiently good hypotheses for them to try to experiment on, what do we need to get to that point? Yes, there's another question.
Yeah, go ahead.
Hey, thanks for this great talk. I just wanted to ask one question on how do you see this being influential in cases like personalized medicine and rare disease medicine?
Yes, absolutely. So I think that there is so much potential here. I'll just start back in RA-PINNACLE for a moment. So one of the things that we're realizing now when where we're able to actually examine these, we have the tools to examine the single cell data from our patients with RA, we can now better disentangle the different kind of populations of cells that are enriched in a particular patient kind of subpopulation of patients. So population is now a little bit loaded, but the types of cell types that we should focus on in a particular subtype of RA, and actually now trace back. I think one thing that I'm really excited about for RA is RA-PINNACLE is being able to now map from the single cell level to the patient level, as in now that we have these, we can make these kind of like patient level predictions about their response to treatment or their stage in RA, but also being able to now trace back to, at the single cell level, what cells were responsible for driving that particular disease or the progression of their disease. And I think being able to now make these contextualized predictions at the cell level and at the patient level simultaneously can allow us to now generate hypotheses that we weren't able to necessarily before. And in terms of this rare diseases, so here I'm really excited to kind of connect what we've done with PINNACLE and Shepherd, so that we can now make predictions about what kinds of therapies, first of all, can we that exists now that we can repurpose for patients with rare diseases. We have a lot of genomic, really fine-grained genomic data for these rare disease patients, because we really have to dig into that kind of resolution to understand what is really causing their disease. And I'm really particularly excited about now being able to trace back, now making patient level predictions, like what disease they have, and being able to trace back to the actual mutations that's causing their disease at the genomic level profile of their disease, and now try to find potential therapies that target that region of the genome, for example. So I think we can really, like I said earlier, with RA-PINNACLE, have this being able to simultaneously model like the genomic level, but also like the single nucleotide level context, but also the patient level context.
Got it. Thanks for the answer.
Thank you for your question. Yes, Matteo?
Hi, Michelle. Thank you for the nice talk. I have a bit naive question, but how do you think PINNACLE and contextualized AI could integrate spatial transcriptomic data? I see that really good fit with the spatial transcriptomic data, the fact that we have the context of the cell.
Yes.
Issue. But in the other hand, we have low level of signals because we have less genes that are measured by spatial transcriptomic. So what is your think about it?
Yes, that's an excellent question. There is a lot of work right now that are connecting spatial transcriptomics with single cell transcriptomics. And I think it kind of follows nicely with the way that we have our networks, right? We have these this metagraph that is organized according to cellular communication and tissue hierarchy, but you can also imagine now actually, incorporating the spatial, because right now it's the network doesn't actually contain the spatial relationships between these tissues, like how far away all these tissues are, or how far away these cells are in the actual space. And so we can certainly, I would be really excited to now incorporate that information into the spatial transcriptomics, from the spatial transcriptomics into this metagraph. I think that would be certainly one place to start to integrate that. Yeah.
I have another question.
Yeah.
I guess you've probably spent a lot of time tuning machine learning models. Do you see a need at that level for improvement, different types of model that scale better, or different optimization kind of strategies?
Are you referring to how can we now, or perhaps maybe can you rephrase your question or restate your question?
Yeah, just in terms of like the ML architectures you're using, do you think
Yeah.
there's some need for improvement there?
I see what you're saying. Okay. Yeah, as I do think that there is a much room for improvement here. I think for as we think about scaling to RA-PINNACLE, for example, I think there are some aspects of the self-attention that is a little bit limiting in terms of computational complexity and computational resources. And so I think there is perhaps room to optimize there as we scale to like from 156 cell types to like 800 cell types, right? Can we do that a bit more efficiently? Right now we have a single unique weight matrix that's cell type specific for every single cell type for each of our 156 cell types. And as we scale to over 800 cell types, that is going to be quite limiting. Now, can we be more efficient in perhaps maybe there's some, we can maybe share some weights or other ways so that we can get around having to have a unique weight matrix per cell type, but still maintaining the cell type specificity or context specificity. Yeah, absolutely.